rounds. Today we have Dr. Isha Jain, who will be speaking to us about turning the oxygen and vitamin dials. Dr. Jain is Assistant Professor of Biochemistry and Biophysics at UCSF. She obtained her BA in Chemical and Physical Biology at Harvard University and completed her PhD at MIT before joining UCSF as a Sandler Research Fellow in 2018. Dr. Jain has received numerous awards over the year, including the NIH Early Independence Award in 2018. Dr. Jain's lab focuses on understanding oxygen metabolism with the ultimate goal of developing novel therapies for metabolic conditions using a combination of genomics, systems metabolism, and animal physiology approaches. So I'll turn it over to you, Dr. Jain. Thank you so much for joining us. If people have any questions, please feel free to put it in the chat or raise your hand, and we can have you ask your question in person.
Wonderful. Thank you, Joyce, for that introduction, and it's a pleasure to be speaking to this audience. So I started at UCSF about 3 years ago, and in general, my lab is interested in understanding how organisms interact with their environment. So how is it that what we breathe and what we eat affects our physiology and metabolism? And then as a consequence, can we change things about our diet or environment as a therapeutic strategy? So rather than just thinking about small molecule treatments, can we come up with creative solutions to different disease states? And we're also moving in the direction of vitamin and cofactor metabolism in a similar setting. Okay, so today I'll primarily be talking about oxygen biology and with a focus on mitochondrial disease, but we're also moving in the direction of cardiac metabolism and physiology. And so we're looking for collaborators and people that are interested in the translational aspects of our work. So it'd be great to talk further. I always like to start with a little bit of a nod to the past, to the first oxygen scientists. And so people have been fascinated by oxygen physiology for hundreds of years, but it was impossible to study the effects of low or high oxygen until the invention of hot air balloons. And so this was the first oxygen scientists. Once hot air balloons were invented, they would ascend rapidly to high altitude, and they would take a cage of pigeons and release a different pigeon at different heights and observe the physiology and survival of these birds. So of course, if the pigeon doesn't do well, the humans will also not fare well. And then unfortunately, there were some tragic accidents. For example, this is a Scientific American article from the late 1800s. It was a flight called the Zenith, and these 3 aeronauts ascended to almost 20,000 feet within less than an hour, and 2 of the 3 perished. So some really sort of dramatic and tragic accidents along the way of studying oxygen metabolism. So luckily, things are more doable in current state, and so our lab has many different types of oxygen setups that are safer and easier to work with. Okay, so the big picture questions we're asking is what happens when there's too little or too much oxygen in the body, and same with the vitamin and cofactor metabolism. And I'd like to provide some context for where we're headed in the future. And we think that ischemic heart disease and myocardial infarction is a particularly interesting setting to be studying these disease states in the future, because it presents a unique situation in which both too little and too much oxygen are toxic. So during the ischemic phase, a lack of oxygen and nutrients results in cell death and pathology. And at the same time, the sudden burst in oxygen during reperfusion is thought to be a primary contributor to organ damage. And at the same time, some of the most robust, at least experimental examples of protection are the situations of ischemic preconditioning and ischemic postconditioning. And so this is really to say that variations in oxygen levels and the timing of such variations can really dramatically affect disease and metabolism. And so can we understand this and use this to our advantage for therapeutic benefit? So we're asking, why is too little oxygen toxic? And of course, this is relevant for stroke, heart attacks, respiratory disease. But surprisingly, we still don't understand the exact metabolic reactions that are failing when there's too little oxygen. So beyond an ATP crisis or energy failure, what else is going wrong? We're also interested in answering the opposite side of the coin, which is relatively understudied, and that is, why is too much oxygen toxic? And so in states of hyperoxia, how is that contributing to disease? How does the body sense variations in oxygen levels? And this is the topic of the recent Nobel Prize for the HIF pathway. If the body can sense variations in oxygen levels, how does it try to adapt in situations like ischemic preconditioning, et cetera? How does the body rewire its metabolism to cope with low or high oxygen? What are the diseases that are characterized by too little or too much oxygen? And then this is really sort of the unusual angle of our lab, which is how can we turn the oxygen dial to low or high as a therapeutic strategy? So how can we modulate oxygen levels for therapeutic benefit? And then we're applying the exact same framework to vitamin and cofactor metabolism. And I'll touch upon that a bit towards the end. Okay, so I'll primarily talk about how we came to find that low oxygen settings are paradoxically a potential therapy for mitochondrial disorders. And then I'll touch a bit about where we're headed in the vitamin metabolism direction. So just a comment is that evolution has really try to solve the problem of having exactly the right amount of oxygen in different tissues. And so, for example, red blood cell formation, our entire cardiovascular system and pulmonary system, and even here on the left corner is an image of E. coli that can sense the amount of oxygen in their surroundings and swim away or towards oxygen gradients. And so there's a lot of beautiful examples of tissues trying to have enough oxygen delivered to their system. And at the same time, there's many examples of cells and organisms trying to avoid excess oxygen. So for example, most stem cell niches are extremely hypoxic. The gut microbiome exists at a very low PO2. And we would like to argue that mitochondria are one of the examples of how the body tries to cope with excess oxygen and detoxify oxygen. So the story began by our focus on mitochondrial disorders. So this was during my training in Vamsi Mutha's lab. And mitochondrial disorders are a really devastating class of diseases. They affect 1 in 5,000 live births. Nearly every organ system is affected, resulting in blindness, deafness, muscle myopathy, neurodevelopmental delay, et cetera. And often children don't survive past the first few years of life, and there's really no treatments that work for these class of disorders. And so I wanted to tackle this problem of developing a novel therapeutic strategy for mitochondrial disorders. And I wanted to do this in an unbiased manner. And so this was right when the first genome-wide CRISPR screens were being invented. And so we decided to use this genome-wide approach and ask, out of all the genes in the genome, which one would be the ideal therapeutic target? So which gene, when perturbed or knocked out, could enable survival in the context of mitochondrial dysfunction and mitochondrial disease? And so cutting a long story short, these were the results of the genome-wide CRISPR screen. So we subjected cells to mitochondrial dysfunction using a small molecule poison of the electron transport chain, which makes ATP in the mitochondria. And then we asked which gene knockouts rescue the phenotype of these diseased cells. And so here are 80,000 dots representing 80,000 knockouts, so 4 knockouts per gene, and ranked by their ability to rescue disease. And so in this top right corner are the knockouts that protect in this disease condition. And surprisingly, all 5 red dots here correspond to the exact same gene, which is VHL, or von-Hippel-Lindau factor. And so this is a really strong signature. Out of 80,000 types of knockouts, the ranks for the knockouts against this one gene were 1, 2, 3, et cetera. And so a really strong signal saying that inhibiting von-Hippel-Lindau factor is protective against mitochondrial disease. And so I first saw this and I panicked because that's a tumor suppressor, right? Why would we want to inhibit a tumor suppressor? But it turns out that this actually pointed to some very interesting biology about hypoxia metabolism. So what is VHL? It's a part of the hypoxia inducible factor pathway. And so a quick introduction to the hypoxia transcription factors. So these were, again, the topic of the recent Nobel Prize. And these are a set of transcription factors that are responsible for an organism's adaptive response to hypoxia. So normally these transcription factors are made in the body, and under high ambient oxygen tensions, they are hydroxylated by these PHD enzymes. And then this hydroxylated form is specifically recognized by VHL, which is the hit of our screen. And VHL is a E3 ligase that marks this protein for degradation. So under normoxia or high oxygen tensions, the HIF proteins are made, degraded, made, degraded. On the other hand, when there's low oxygen, this cascade does not take place. HIF enters the nucleus and it turns on transcription of several hundred genes. And these genes involve things like upregulating anaerobic glycolysis, increasing hematocrit, VEGF, vascular density. So the body's way of trying to increase oxygen delivery or rewire metabolism to bypass oxygen needs. And so what we had found was that knocking out VHL was protective in mitochondrial disease. So what happens when you knock out VHL? So VHL is the negative regulator of the HIF response. So if you knock VHL out, you're essentially tricking the body into thinking it is hypoxic. And so even in a normal oxygen environment, you're triggering the adaptive pathways for low oxygen. And so in some sense, this makes sense, right? Hypoxia is a type of mitochondrial dysfunction that evolution has tried to solve the problem for. It's an adaptive program for impaired oxidative metabolism. On the other hand, it's a bit strange because hypoxia is thought to be a bad thing. And so how is it that this is protective? So we charged along. We wanted to see if we could target this pathway for therapeutic benefit. And it turns out there was a drug that was being developed at that time, and now it's actually approved in certain countries, that inhibits the other negative regulator in this pathway. So it has the same effect of activating this HIF response. And we were able to show that this is protective in cells and in zebrafish. And so here are growth curves of cells exposed to different types of mitochondrial disease. So as a reminder, in the mitochondria, the electron transport chain is made of these 5 complexes, 1, 2, 3, 4, or 5. And in patients, you can have dysfunctions of any of these different complexes. And so if we poison any of these complexes, 1, 3, or 5, you impair cell growth in red. And now if you activate this hypoxia response with this drug that I just mentioned, you almost completely, if not completely, rescue this growth defect in blue, right? So activating the hypoxia response has this protective effect across many different types of mitochondrial dysfunction. Okay, so this is all cell culture, and then we showed that in zebrafish as well. But in the end, we really want to focus on mammalian models of disease, and that's really where the therapeutic benefit would be apparent. And so I spent a fair amount of time trying to optimize this drug. Turns out it was being developed for renal anemia, and so it was really not having any pharmacokinetic sort of benefit for the brain. And so I struggled for a year trying to get this across the blood-brain barrier, didn't have any luck. And then this was sort of a late night experiment that I had a friend across the street in Warren Zapol's lab. And Warren Zapol, for those of you that know, was the inventor of inhaled nitric oxide for blue baby syndrome. And so that lab really studied a lot of gas physiology. And so I had this friend across the street, and I asked this friend, can we borrow your hypoxia chamber, right? We're trying to activate this hypoxia response. It's not working with drugs. This is probably not going to work, but can we just sneak in a cage of our mice into your hypoxia chamber and see if that can activate the hypoxia response? So it turns out this was the magical moment in this project. So I'm just going to show you a video first. So these are mitochondrial disease mouse models. So this is a mouse that lacks complex 1 of the electron transport chain. And this is an MRI of the mouse, and you can see there are these lesions in the vestibular nuclei. And this is exactly what happens in the human patients that have what's called Lee syndrome, which is the type of mitochondrial disorder caused by complex 1 deficiency. And these mice are very representative of the human disease. So they have these lesions, they have body weight loss by about 2 months of age, they're blind, they're deaf, they have locomotor defects, and they ultimately succumb to respiratory failure due to these lesions in the vestibular nuclei. And so we took these animals and we placed them in these hypoxia chambers, and this was the effect. So these 3 animals in the middle. So first of all of these mice have the genetic mutation. They all have complex 1 mitochondrial disease. They're all siblings. It's a very well-controlled experiment. And the 3 in the middle have been living at sea level, or 21% oxygen. And the other 2 have been living in hypoxia, so about 5,000 meters altitude, a level that's pretty high altitude, but there are cities that exist in Peru and Nepal at that altitude. And so there was this really magical effect where the animals that were at sea level, so San Francisco, Boston, New York, where a lot of us have spent a lot of time, they were really, really sick, minutes away from death. Whereas the other two, the only thing we'd done to them was chronically expose them to high altitude or hypoxia. And it was this really dramatic phenotypic effect where these animals were completely indistinguishable from healthy wild type animals. And so this is what started our journey towards developing hypoxia as a therapeutic strategy. So this is a Kaplan-Meier survival curve of these mice. And so the knockout diseased mice in room air or sea level died about 2 months of age. When we first published the study, then all of the mice that were diseased but in hypoxia were all alive. And it turns out it's about a 5-fold extension in lifespan. So really dramatic therapeutic benefit just by putting these animals in low oxygen. And we could rescue every aspect of disease. So in red here, these mice gradually lose weight and die at sea level. This is rescued by hypoxia in blue. These mice have a dramatic drop in body temperature in room air in red, and this is rescued by hypoxia in blue. We can measure their ability to stay on a rotating rod, which is impaired in the Normoxic knockout mice and rescued by hypoxia. And really every aspect of disease was rescued, from neuropathology to behavior to disease biomarkers. Okay, so this really opened a sort of range of new questions that we wanted to answer and are still really chipping away at within the lab. So what is the mechanism of the hypoxia rescue? If we can understand that, perhaps we can directly target that mechanism rather than moving to high altitude. What is the underlying pathophysiology of disease at different oxygen tensions? So is the cause of death the same in hypoxia eventually when they do die, or is it a different organ system that is failing? Can we reverse disease in addition to preventing it? And this is really relevant from a therapeutic perspective. Can we actually have a patient come in that's already had a metabolic crisis and reverse the symptoms? And then, again, we can make our house hypoxic. Turns out you can make a room hypoxic for a few $100s. There's machines that the athletes use when they're training for marathons to make your house hypoxic. But ideally, we would have another solution instead of living in hypoxia. And then importantly, does this concept apply to additional disorders beyond mitochondrial disease and perhaps more common conditions, neurodegenerative or other organ systems? Okay, so our first question was, what is the mechanism of rescue and what is the underlying cause of pathogenesis? And so suffice it to say that we haven't completely answered this question yet, but we have some early clues. And so this was actually Dr. Zapal's idea, where we took our animals that had mitochondrial disease genetically, and then we did a very simple experiment where we put them in hyperoxia. So we turned the oxygen dial in the other direction, very moderate supplemental oxygen, moderate from sort of a mouse perspective, which is 50% oxygen. So wild type animals are completely benign at this oxygen tension, but look what happens to the mitochondrial disease mice. So within 2 or 3 days of relatively moderate hyperoxia exposure, the animals crash. So they abruptly lose weight and die. And so this is really sort of puzzling and has some pretty dramatic clinical implications, which is if you have mitochondrial dysfunction, genetic or otherwise, perhaps we should be a bit more cautious about using supplemental oxygen. And we had an interesting sort of anecdotal stories where we had a clinician call us after this initial story where he had a patient come in that had mitochondrial disease and they thought, oh, broken mitochondria, let's give it more oxygen, right? And so they gave the patient hyperbaric hyperoxia, thinking, oh, we'll push the mitochondria harder. And that patient was comatose within 24 hours. And another story where a patient came in with Leber's hereditary optic neuropathy, was blind in one eye, and came in, was given hyperbaric hyperoxia, and then within 24 to 48 hours was blind in the other eye. So these are anecdotal stories, but they're in line with our sort of well-controlled animal experiments, suggesting that at least when it's not clinically indicated, we should be a bit more cautious about supplemental oxygen. And this is talking about the rare mitochondrial disorders, but I think there's a growing appreciation for this concept more broadly about the toxicities of supplemental oxygen. And this is something that we're pursuing sort of vigorously in the lab right now. Okay, so in terms of the mechanism, this pointed us to two different sort of categories of hypotheses. So one option is that low oxygen is a signal, right? And the way that I was describing earlier, where it triggers this adaptive program through transcription factors, et cetera. So a metabolic rewiring that's beneficial in the context of mitochondrial disease. And then the other bucket of questions is that perhaps low oxygen is simply a way to decrease the production of reactive oxygen species. And this is, a very general term, but perhaps you have less superoxide, less hydrogen peroxide if you have less oxygen, and that's the relevant variable. So we're still chipping away at it. I think we're leaning more towards the right side of this chart. To answer this question, we returned to our original hypothesis. So as I mentioned, we came to this story by activating the HIF response, but we were never able to directly test whether this HIF response was protective in mitochondrial disease in the animal model because of this issue of getting the drug across the blood-brain barrier. So we finally, decided to bite the bullet and do the genetic crosses. And so we crossed our mitochondrial disease mice with mice that lack the negative regulators of this pathway. So activating the hypoxia response in Normoxia in these diseased mice. And so we actually, much to our surprise, saw the completely opposite effect. So the double knockouts are in red and the single knockouts are in black. And so it turns out that genetically activating the HIF response or hypoxia response is not protective in an animal. It's actually the opposite. If anything, it's detrimental. And we think it might be detrimental because part of the hypoxia response is to increase oxygen delivery, right? Increase vascular density, increase erythropoietin, increase hematocrit. And so you might actually be causing tissue hyperoxia in this context. Okay, so this is a bit shocking and disturbing because we came to this whole concept by the HIF pathway, but it turns out in vivo, it seems to not be about this adaptive program, and perhaps it's about oxygen itself. Okay, so what is happening to oxygen in these mitochondrial disease mouse models? So we first measured whole body oxygen consumption in these disease mice and in control animals. And you can see as a function of age, the wild type or healthy animals have a baseline amount of oxygen consumption. Interestingly, if you have mitochondrial disease or dysfunction, your whole body oxygen consumption goes significantly down. And this makes sense because about 90% of oxygen that we breathe is directed towards the electron transport chain of the mitochondria. So even at the whole organism level, if you have mitochondrial dysfunction, you have much less whole body oxygen consumption. But then this brings us to the question of supply and demand, right? So we're breathing the same amount of oxygen, but if you have mitochondrial disease, you're consuming less oxygen. So what's happening to the local PO2 in the brains of these animals and these patients? So we can stick a Clark electrode, an electrode into the brains of these animals and measure the oxygen concentration or partial pressure of oxygen in the diseased versus the healthy animals. So if we just look here at an FiO2 of 0.21, so normoxic sea level breathing, you can see that wild type animals or healthy animals have this amount of oxygen in the brain. Now, if we take the matched mitochondrial disease mice for the same amount of inhaled oxygen, their brain has double the amount of oxygen. So it's essentially like having pockets of hyperoxia in the brains of these mice. And so this is really a new perspective because we've never really thought about diseases of hyperoxia. So it seems like mitochondrial dysfunction might be characterized by this excess oxygen phenotype. And interestingly, when we put these animals in hypoxia, so an FiO2 of say 10% oxygen, it takes this hyperoxia in the tissues and brings it back down to wild type levels. So maybe hypoxic breathing is working by just normalizing this excess oxygen in the brains of these mice. And so we're starting to try to chip away to answer why is oxygen toxic? And so we've done some preliminary experiments, and this is taking mitochondria isolated from the knockout in the wild type mice, and then as a function of oxygen, measuring the amount of hydrogen peroxide, which is one type of reactive oxygen species. And you can see that there's this clear linear relationship between amount of oxygen and hydrogen peroxide produced. And that relationship is the same, in this case at least, in the diseased and controlled mice. mice. But as I told you, the knockout mice have double the amount of oxygen in the brain. So they might have double the amount of hydrogen peroxide or superoxide in the tissue level. And so we have a lot of projects in the lab ongoing trying to understand exactly which pathways are being disrupted in this situation of excess oxygen. Skip that. Okay, so what is the underlying pathophysiology of disease? What is the cause of death in different oxygen tensions? So this is the final survival curve once this experiment was finished. So in room air, they died about 2 months. In hypoxia, they died about a year, a little bit less than a year. And in hyperoxia, they die within 2 or 3 days. So initially we thought, oh, they must be dying of the same cause in all 3 oxygen tensions, right? We're either preventing or accelerating death, but presumably the cause of death is the same. Turns out that's not the case, surprisingly. So in Normoxia mice, if we do an MRI right before death, you can see these lesions, these T2 weighted hyperintense lesions in the vestibular nuclei, these 2 white dots, and in the olfactory bulb. Now, if we do MRIs in the mice right when they're at the brink of death in hypoxia or hyperoxia, these lesions are not there. And so surprisingly, the cause of death might actually be completely different as a function of oxygen tension. And so, we didn't go too deep into this, and we probably should at some point in the future. But if we take the mice in hypoxia right when they're at the brink of death at about a year of age, they almost had the sudden death phenotype where they would just drop dead. And so we thought, okay, maybe it's some sort of a cardiac issue. And so we saw some early signs of left ventricular hypertrophy in these knockout mice right when they were about to die in hypoxia. And we also saw some evidence of arrhythmias, which we never followed up on. But it might be that in hypoxia, the cause of death is actually cardiac. Interestingly, in hyperoxia, it seems to be a completely different organ system. And what we noticed was that in the knockout mice exposed to hyperoxia, they developed very severe pulmonary edema. And so in 3 different oxygen tensions, there was 3 different causes of death. Normoxia was neurodegenerative. In hypoxia, it was potentially cardiac. And in hyperoxia, it was pulmonary oxygen toxicity. And so this is what got me really interested about trying to understand oxygen physiology at the whole animal level, right? Perhaps different organ systems and different tissues have different set points for oxygen toxicity, where at one oxygen tension, one organ system fails and another at another oxygen tension. Okay, so as I mentioned, a lot of these experiments were done in an effort to prevent disease progression, but typically mitochondrial diseases are not included in prenatal testing, and often patients will be diagnosed after they've already had a metabolic crisis. And so we wanted to understand if we could reverse disease in addition to preventing it. So we can take these knockout mice, we can leave them in room air in yellow, and they gradually lose weight and die. We can start hypoxia at an early stage before disease, and they gain weight and do okay. Now we can start another cohort of animals where we let them lose weight. They're at the brink of death, maybe a day or 2 away from death, and we turn the oxygen dial to low. And there's this like almost magical reversal of disease phenotype. So within 24 to 48 hours, you can see this upward swing in the disease trajectory, and the animals start to reverse disease. And perhaps the most exciting part was that we could actually reverse the neurological lesions. And so we can perform MRIs in these mice at the late stage of disease. You can see the 2 dots in the vestibular nuclei. And we can take these same animals, put them in hypoxia, and scan them again a month later, and the lesions have disappeared. And more recently, we could even do this in 1 week of more extreme hypoxia. So very sort of rapid and dramatic reversal of neuropathology and every other aspect of disease. Okay, so that's great, but now how do we turn this into a practical therapy? And so can we find a way to cause tissue hypoxia without living at high altitude? And so we had fun coming up with creative ways to cause tissue hypoxia, some of which are more practical than others. So you can live at high altitude, that's one solution. We would never do this, but theoretically you could occlude the carotid and cause brain hypoxia that way. Bloodletting is used in current days for polycythemia and other conditions. So theoretically, you could basically cause anemia. Low dose carbon monoxide is actually being used for certain inflammatory conditions. And so theoretically, you could use low dose carbon monoxide to compete with oxygen for hemoglobin binding. And the most practical and the one that's most likely to actually reach the patient population is left shifters. And so they're small molecules that are being developed primarily for sickle cell anemia that affect the binding affinity of hemoglobin for oxygen. And so releasing less oxygen into the tissues. And so you can use these drugs to artificially cause tissue hypoxia. And so that's, we're working on this class of different types of perturbations. So just as a proof of concept, we wanted to see if we could hit the oxygen delivery equation in multiple different sites and see if that was protective. And indeed, low dose carbon monoxide or anemia remarkably extends lifespan in these diseased mice in red compared to untreated mice in black. So again, we're not going to be necessarily proposing these, but just as a proof of concept. And so ultimately, again, the left shifters are the most likely to reach the patient population. Okay, so this is all about this mitochondrial disease with a mutation in this complex 1 subunit, but is this relevant for other genetic disorders and perhaps even other common conditions? So more recently, I had performed a genome-wide CRISPR screen looking at which genes are essential as a function of oxygen tension. And this is relevant beyond the concept of hypoxia therapy, right? So there's so many different conditions that are characterized by low or high oxygen. So what genes are selectively essential in hypoxia or hyperoxia? And so to do this, we can create this genome-wide pool of knockout cells again, to knock out every single gene in the genome, and then subject it to low, medium, or high oxygen, and ask which genes are needed as a function of oxygen, and importantly, which disease genes cause a fitness defect in normoxia, but is rescued by hypoxia. So this is a way to ask genome-wide, what diseases can we target with hypoxia therapy? And when we look at which gene categories are selectively essential as a function of oxygen tension, reassuringly, we get the entire electron transport chain, which is denoted as OXPHOS, as being selectively needed in normoxia, but less needed in hypoxia, which is in line with with the work that I just described. And interestingly, we also got this category of hits on the other side of the coin, which is genes that are needed in low oxygen, but not high oxygen. So these would be genes that are more essential in the context of ischemia or hypoxia. And surprisingly, these are all related to this sort of neglected part of the cell called the peroxisome. So this is an organelle in the cell that is involved in lipid metabolism, reactive oxygen species, et cetera. So more to come on that in the future. But again, getting back to this question of what are the prime disease candidates for hypoxia therapy, right? Can we extend this concept out to a broader set of disorders? And from this screen, we actually nominated an additional 75 monogenic disorders that could be rescued by hypoxia in our cell culture setting. So these are 75 disease genes that were hits in our screen. And a lot of them are mitochondrial. So in dark red are mitochondrial electron transport chain genes. In light red are mitochondrial disease genes that are not clearly linked to mitochondrial electron transport chain. And interestingly, there's some here related to aging and Parkinson's disease. And then there's a whole class of non-mitochondrial genes that are also being rescued by hypoxia. And so we're working hard in the lab to try to understand if these animal models of disease can also be rescued by hypoxia, again, extending our findings to a much larger patient population. Okay, I'm going to skip this for now. Another place where we can look for clues as to which diseases can be rescued by hypoxia is epidemiological clues. So are there certain diseases that are at lower incidence at high altitude than in sea level patient populations? And interestingly, many of the cardiovascular disease risk factors and things that are involved in metabolic syndrome are ameliorated at high altitude. And so this is a very robust finding in mice and humans in our hands, and has been observed in soldiers at high altitude for quite some time, that there's a dramatic decline in obesity and body weight loss at high altitude. So I think this is mice exposed to hypoxia, they lose about 15 to 20% body weight over the course of a month. And this is thought to be related to a neurohormonal response to hypoxia. And so it's really food intake that's decreased. And so perhaps there's some link here to food intake and hypoxia therapy. Interestingly, serum cholesterol levels decline very dramatically at high altitude. And so this is a plot from a study in the Indian Army where they took 10,000 soldiers and left them at high altitude, 10,000 at sea level, and measured various physiological parameters. And they saw this time-dependent decrease in serum cholesterol. And interestingly, this can also be mimicked by this drug that I was telling you about that activates the HIF response, where if you give this drug, there's a decline in total cholesterol levels. And if you stop the drug, there's an increase in cholesterol levels. And so the mechanism here is not known, but perhaps hypoxia or the hypoxia response is protective for these different sort of cardiovascular risk factors, disease risk factors. And I didn't mention it here, but serum glucose also is dramatically affected, where glucose levels drop by 2x or 4x in mice when they're exposed to hypoxia. Okay, so where are we headed with this? We started a phase 1 clinical trial. This is over MGH, where we took healthy volunteers and left them in high altitude tents for a week. And of course, they're fine because people have lived at high altitude for generations. But it's nice to be able to do this in a controlled setting. And so we're hoping that, in the near future, we can really translate this to the patient population and hopefully using small molecules such as left shifters. So for this part of the talk, in summary, hypoxia extends life of a mitochondrial disease model by 5-fold. We can reverse disease in addition to preventing it. Activating the HIF response is not sufficient to rescue mitochondrial disease. It might be more relevant for other types of conditions like decreasing cholesterol levels. Then mitochondrial disease animal models and patients as well are characterized by excess oxygen in their tissues, and perhaps hypoxia is just normalizing this excess oxygen. We can come up with creative ways to decrease tissue hypoxia, and more to come on that in the future. And now we've nominated 75 monogenic disorders that can be rescued by low or high oxygen. Okay, so I just told you about mitochondrial disease and turning the oxygen dial as a therapy. And now we have part of the lab that is trying to translate this concept of turning the metabolic dial to a completely different context of vitamin and cofactor metabolism. So this is still early days, but I'll just give you a clue of where we're headed. And if anyone has any ideas to work together in this space, that would be great to hear. So from the late 1800s to the mid 1900s was this beautiful era of biochemistry. So the 13 vitamins were discovered in this time period, and over a dozen Nobel Prizes were awarded for the so-called vitamin hunters. It's a really beautiful era of biochemistry where the function of these vitamins was studied, and the effects of vitamin deprivation were studied in animals and humans. And then something happened around 1950s with the invention of molecular biology, which is vitamins entered this sort of pseudoscience supplement type of realm. But that being said, there are many case reports where variations in vitamin levels can really have a dramatic effect on disease. And so vitamin supplementation in certain cases with certain genetic disorders can be really beneficial. And historically, for example, niacin was used as a way to treat dyslipidemia. And so there is clear evidence that in certain cases, certain vitamin levels are affecting disease progression. And in fact, there's over 5,000 clinical trials using variations in vitamin levels, but oftentimes the basis of these trials is not rooted in rigorous scientific framework. And so it's someone noticed that variations in B6 levels affect some particular disease and a clinical trial was started. But can we take a more sort of rigorous approach to nominating diseases for megavitamin therapy or variations in vitamin levels? And of course, from a public health perspective, vitamin deficiency affects a very large part of our population. So 250 million preschool children are deficient in vitamin A, leading to blindness in some cases. Much of the aged population is deficient in vitamins, and this is despite adequate dietary intake, there's problems with gut absorption. And so are these subclinical levels of vitamin deficiencies affecting chronic conditions like diabetes and metabolic syndrome? And so can we understand which vitamins, when subclinically deficient, affect which disease states, and understand the mechanisms behind such disease pathologies? So we're asking which diseases can be treated by megavitamin therapies? What are the causes and consequences of subclinical vitamin deficiencies? And can we target these for certain chronic conditions? Surprisingly, we still don't know the functions of many of the vitamins in the body. And so, for example, vitamins A, C, D, E are often called antioxidants, but that's not a clear biochemical function. So what exactly are they doing in the body? And we're using a host of approaches similar to what I described, from animal physiology to developing, hopefully one day, novel vitamin reporter tools to genome-wide CRISPR screens to try to tackle these questions. And I think I will actually stop there. So first of all, a huge thank you to Vamsi Mutha and Warren Zapal, which is where a lot of this earlier work was done. And actually Warren Zapal was one of my closest mentors who recently passed away. And so we miss him and hope we can carry on his legacy, continuing to work on oxygen physiology. And thank you to my current lab, and also a shout out to Rahul Goli, who did a lot of the earlier work, who's currently a UCSF cardiology fellow, and Alan Beck, who's currently in my lab working on oxygen toxicity. So I'll stop there and take some questions.
Thank you, Dr. Jain. That was wonderful. Does anyone have any questions? Feel free to unmute yourself or raise your hand and we can ask your question in person. I guess I was wondering, are there specific physiologies, especially within cardiology, that you're interested in looking at or targeting next with a lot of this work that you're doing?
Yeah, it's a great question. So I think the two category of cardiovascular related disorders that we're thinking about is ischemia reperfusion injury, as I'd mentioned. So we're doing a lot of genome-wide CRISPR screens and iPSC-derived cardiomyocytes, trying to look at factors that are protective in ischemia and hypoxia. And then I think also sort of metabolic syndrome and dyslipidemia is another category where we're trying to understand interactions between oxygen and diet. Okay, if anyone has any other questions or ideas, please feel free to email me. I'd love to brainstorm further.
Thank you so much. Thanks for joining us, Dr. Jain. Hopefully next time we can do this in person. And thank you everyone for joining us for our cardiology grand rounds. I'll see you guys next week. Okay.